Hi, in our last video we talked about how coloring cell specimens can be done through two processes. They were immunohistochemistry and immunofluorescence. Now, two of the most prominent types of dyes used in immunofluorescence include the DAPI dye and the Ki67 dye. And over the course of two videos, we're going to explore the two dyes. Now, both have really different purposes, but are very equally important biomarkers, specifically in the detection and treatment of cancer, so please do stay tuned. Now we're going to start with the DAPI dye here. Now its full name is 4,6-diamidino-2-phenylindole, but we won't be calling it that. We're just going to keep it short and simple as DAPI. But to understand how the DAPI dye works, we need to first understand the structure right here. Now you're probably more or less familiar with the structure. It's the DNA double helix structure. Now DNAs are in all of our body cells, with the exception of red blood cells and cornified cells, and are comprised of base pairs of adenine and thymine, and guanine and cytosine. Now, the adenine and thymine pair, normally referred to as the AT pair, comprise of two hydrogen bonds, as illustrated in the image right here, and the guanine and cytosine pair, normally called the GC pair, comprise of three hydrogen bonds, as illustrated in the image similarly. Now, to kind of make this discussion pertain to the DAPI dye, the DAPI is a fluorescent dye that binds to the AT pair of the DNA to emit sort of like a neon bluish light. Now, the reason why this emits a neon bluish light is because when the DAPI binds to the pair, it absorbs light of around 358 nanometers of wavelength and kind of emits a blue cyanish color. So in regions where there are a lot of cells, like a high concentration of DNA and AT pairs, it kind of appears in this neon blue color. But why is staining DNA so important? Well, it's important because it can be a very important biomarker for cancer.